My name is Filtration and today I am here to present my skill on the topic filtration methods used in the downstream processing. So first let's understand what is downstream processing. So basically downstreaming processing refers to all the process carried out after fermentation or the cell culture to recover and purify the desired biological product. The objective is to obtain a product with a high purity while maintaining its biological activity. Downstream processing consists of the four steps. First, removal of insoluble material, product isolation, purification and formulation. Filtration is the first separation step. During fermentation, the broth contains microbial cells, cell debris, nutrients, proteins, and other impurities. Filtration removes this unwanted product for further purification process such as chromatography and crystallization. If we talk about the principle of filtration, so the principle of filtration is based on size exclusion. A filter contains tiny pores that allow liquid and dissolved molecules to pass while retaining larger suspended materials. The movement of liquid through the filter is produced by the driving forces such as gravity, vacuum, positive pressure, and the centrifugal forces. Gravity that relies on the weight of the fluid column. Vacuum pressure differential created below the filter. Positive pressure applied above the filter medium, and the centrifugal force, rotational force for rapid separation. If we talk about the factors affecting filtration, then there are many factors that affect the filtration process like particle size, membrane pore size, pressure difference, viscosity, temperature, concentration, and the nature of filter medium. How the particle size and shape affect the filtration? By determining which particles are retained. Then membrane pore size define the cutoff for the separation. Temperature influence viscosity and membrane performance. Now let's understand what are the types of filtration. There are two types of filtration, depth filtration and the surface filtration. The first is the depth filtration. In this method, suspended particles are trapped inside the filter matrix. The filter consists of several layers, allowing particles of different sizes to be captured. It has a high dirt holding capacity and is commonly used for the clarification of fermentation broth. Now, what are the examples of the depth filtration? Cellulose filter, glass fiber filter, diatomaceous earth filter. Advantages of depth filtration like they handle high particulate load, long filter life, low pressure drop, and the limitation difficult to sterilize completely, low filtration percent. The second is the surface filtration. Here, particles are retained only on the surface of the membrane. As filtration continues, a filter cake form increases filtration resistance. Surface filtration is commonly used when sterile filtration is required. Then examples of surface filtrations are membrane filtration and the screen filtration. Advantages: high filtration accuracy suitable for sterile filtration. Limitation: membrane falling occur rapidly, low dirt holding capacity. As we discussed above, this is the diagrammatically representation of depth filtration and the surface filtration. Now, if we talk about the techniques, and the first technique is membrane filtration technique. Membrane filtration technique is classified according to the membrane pore size. Microfiltration has the largest pores and remove the microbial cells, yeast and the suspended solids. Then second, ultrafiltration has smaller pores and is widely used for concentration and purification of protein, enzymes and the antibodies. Then the third, nanofiltration, which remove the viruses, small organic molecules and the divalent ions. Fourth, reverse osmosis has the smallest pore size and is mainly used to produce pharmaceutical grade purified water by removing salt and the dissolved impurities. Now the second filtration method is cross flow filtration, which is also known as the tangential flow. How it works? In cross-flow filtration, the feeding solution flow parallel to the membrane surface rather than the perpendicular to it. The fluid passes through the membrane while retained particles continue following across the surface. This tangential flow continuously sweeps the membrane surface, preventing the buildup of a thick filter cake and maintaining consistent flow rate and extended operation period. Then, cross-flow filtration is extensively used in a modern biotechnology industries for purification of vaccines, monoclonal antibodies, proteins, and the enzymes. Advantages of this technique is to reduce membrane fouling due to continuous surface sweeping, continuous operation capacity, high product recovery, increased membrane lifespan, highly suitable for large-scale industrial production. Now, let's understand what are the advantages, limitations, and their applications of the filtration. So, filtrations offer many advantages. It provides efficient separation, maintains sterility, can be easily automated, and is suitable for large-scale industrial production. However, it also has limitations like membrane fouling is the major challenges because particles block the membrane and reduce the filtration efficiency. Concentration, polarization and membrane replacement also increase the operational cost. Despite this limitation, filtration remains one of the most widely used separation techniques in pharmaceutical, biotechnology, food and beverages, and water treatment industries. Industrial applications of filtrations, as I talked already about that. So, filtration technology introduced to wide range of industries where product purity, sterility, and efficient are paramount. The following sectors are pharmaceutical industry, food beverages, biotechnology industry, dairy industries, and vaccine manufacturing industry, enzyme production, antibiotic purification. Last but not the least, I conclude that filtration is an essential unit operating system in the downstream processing because it removes the impurities while preserving the quality of biological product. Different filtration methods are selected according to the particle size, product characteristics, and industrial requirements. Among all the filtration techniques, cross-flow cross-flow filtration is most preferred because it minimizes membrane fouling and provides high product recovery. Therefore, filtration plays a vital role in ensuring the safety, purity, and the quality of biopharmaceutical products. These all are the references from which I have cited my content. So, thank you. Hope you like